we'll get into that. Yes, sir. Fine. Now it's all right. We are connecting to YouTube, sir. That's what.
Yes.
Another 2 minutes.
It is telling meeting is now streaming live on YouTube.
Yeah, correct.
Sir.
Venkatesh?
இந்த ஆன்ல இருக்கு விஜய். விஜய் உன் ஆடியோ ஆன்ல இருக்கா? ஆஃப் பண்றா.
Andiran has also joined.
Yes, sir. Yes, sir. Venkatesh?
Yeah.
Yeah. Start பண்ணலாம். That's what I call you. Okay. We'll start?
Sure. Go ahead.
Oh, okay, sir. Dear participants, good evening to one and all.
Sir.
Yes, it's audible.
No, see, but you are not visible.
Visible, sir.
Yes, sir.
Because now the screen of Murli is only visible.
No, sir, it is the side, it was visible, sir, for everybody.
Separate box is available.
But now you should come to the focus.
Yeah. Venkatesh? Start பண்ணிடலாம். Start பண்ணது down பண்ணிடுங்க.
சரி, okay. Thank you. இது download start. Okay, thank you. Start பண்ணு. Okay, bye.
Dr. Murali?
Yes, sir.
You just after introduction you can chat.
Audible, sir?
Before that you can.
Hello.
No, change the screen. Now let Dhanapal should come to the screen.
Screen is okay now?
Yeah.
No, but see the main screen. You should come now when you are talking.
Yes, sir.
Now is our Murali Krishnan's screen is only available.
Yes, sir. Already it was shared by them. Venkatesh?
Audible?
Audible. Screen share வந்து காமனா வரும்னாரு. Slide அப்புறம் பண்ணிக்கலாம்ல sharing of screen.
இல்ல அது ஸ்பீக்கர் வியூ போயிடும். அது போயிடும் ஆட்டோமேட்டிக்கா போயிடும்.
Okay. Sir, we'll start, sir.
Okay.
Okay. Once again, good evening to one and all. Dear participants, I am very much happy to invite you for this 7th international webinar, which is organized by Department of Pharmacy, Annamalai University, jointly with Annamalai University Pharmacy Alumni Association, which is sponsored by Aries Formulation Private Limited and co-sponsored by Eastmed Life Sciences. Today, once again, we have distinguished alumni, Dr. Mullakrishnan Dhanasekaran, Professor of Pharmacology from Auburn University, USA. Already it was time little bit late, so I now request our head of the department to say a few words about this seminar, this webinar. Please, sir.
So, good evening, friends, all the participants. Good morning to Dr. Murali Krishnan. And this is a great occasion that we are able to hear him, Murali, after a long time. And see, if you look at last decade or so, our Indian system of medicine has given due importance. Earlier, even the Ayush was not a ministry. It was just a component of the Ministry of Health and Family Welfare. Now, Ayush becomes an independent ministry. And I have my own experience of how the Ayurvedic science works. We conducted a research work. Both facilities are available. We made available in All India Institute of Medical Science for arthritis treatment. One side we had the allopathic, and the other side we have the Ayurvedic clinic. And it is the choice of the individual patient to go. And at the end, we compared. There is no difference as far as the outcomes are concerned, whether they have gone with the Ayurvedic treatment or traditional treatment and the treatment offered by the allopathic doctors. So that made me to understand that how much potential we had prior to being in the modern allopathic science students. I never imagined that we had so much potential, and I started believing since then. Now we have our Murali Krishnan, who is going to compare, probably validate how the Ayurvedic treatment protocol in terms of modern pharmacology. I'm sure we all are going to get benefited from his talk. Okay, from my side, Dhanapal.
Yes, thank you, sir. Now I request Dr. Venkatesh to introduce our today's speaker, Dr. Murali Krishnan Dhanasekaran. Please, Venkatesh.
Sound is not audible.
If now it's audible.
Yes, Venkatesh.
Yes, now
Oh,
audible.
yes, yes.
So very good evening to all. This is our seventh.
Sorry.
This is a webinar, international webinar series Department of Pharmacy organizing. I am very happy to introduce Dr. Murali Krishnan sir for as a today's speaker. I'm very happy. Sir, Murali Krishnan sir is completed B Pharm from Annamalai. It's our proud. He is a Annamalai University alumni. He completed B Pharm.
Yes.
What happened?
Yes. Yes.
Sir? Sir?
Yeah, talk.
Yes, Venkatesh, go ahead. Okay.
Hello.
Okay, Venkatesh.
Hello.
There is a
Yes, Venkatesh. Okay. Dr.
Hello.
Murali can start, Murali.
Good evening, everyone. I'm
Good
going to talk about. Good evening, everyone. I'm going to talk about validating Ayurvedism, and I picked two complicated disorders. One is a movement disorder. Other one is a mental disorder. So let's go step by step, and then we'll see how Ayurvedism can be validated. And as Dr. Mohanta was telling, how can we compare with the existing pharmacological drug? So the whole thing is about how we can prove that Ayurvedism is equal or better than the existing pharmacological therapy. That's the concept that we are going to look at. First and foremost, I have no words to express my thanks and gratitude. It's all, my life started from Annamalai University, where I am. My journey, my scientific journey, my personal journey, everything started from Annamalai University. It's the true words that's coming out of my mouth. So I would like to sincerely thank and dedicate the whole thing to my faculty, the staff, my seniors, my classmates, and my juniors. I really sincerely thank every one of you. So the journey started from Annamalai University. Thank you. Thank you, everyone. So today, I'm going to just start off a brief of what is Ayurveda. Then let's go into neurodegeneration. So we are talking about movement disorder, mental disorder. So both the disorders are associated with the cell nervous system. But let's find out the word, what is neurodegeneration? Then we are going to look into two pathological conditions. Associated with movement disorder, I'm going to look into Parkinson's disease. And then with regard to mental, we are going to look into a memory disorder, which is Alzheimer's disease. And with that regarding, how does Ayurvedic botanicals play a role with regard to preventing the neurodegeneration? For Parkinson's disease, I'm going to look into Mucuna pruriens. And for Alzheimer's disease, I've done two drugs, but I would like to focus on Centella. We have first published on Bacopa maniera, but then we later published on Centella asiatica. But I have done a lot of work with Centella asiatica with modern tools and technology. So that's the reason I'm going to focus on Centella asiatica. So now, that is the introduction. So we are going to intellectually look into Ayurveda, neurodegeneration, two major pathological conditions, not mental, two major neurodegenerative disorders. One is Alzheimer's, other one is Parkinson, and two botanicals, how they can reduce the neurodegeneration. So as you all know, when we say Ayurveda, the word Ayu comes from life and Veda is science or knowledge. So Ayurveda is nothing but the science of life, the medical science of life or the knowledge of life, however way you want to look into. So that is Ayurveda. So I know there are a lot of students among us. So to start with, whenever we look into a pathology, try to understand what is the anatomy associated with that. Then now, so whenever you think about a pathology, is it a problem with regard to anatomy or the function, the physiology, or both? So whenever we think about a pathological condition or a disease state, we have to start our focus from the structure and the function, anatomy and the physiology. So we have to be very strong when we talk about pharmacy, when we talk about we as in healthcare, we have to understand the concept basically from anatomy, physiological aspect, and what is the change that is occurring. Do you have a change in the structure or the function that is associated with the pathology? And then how can you overcome the existing problem, which means how can you treat? So whenever we think about treatment, so as a pharmacist, you guys have to understand that you can have a prophylactic therapy or you can have a preventive, prophylactic or a therapeutic approach. Okay. So now we think about pharmacology. Again, as a pharmacist, the most important thing for us is the adverse drug reaction. How as a pharmacist, we understand the adverse drug reaction, how we can formulate various formulation to overcome these pharmacological deficiency or adverse drug reaction. The whole aspect is about patient care, how we can improve the healthcare in the society. That's the whole approach. So we all have entered this particular field, a holy field, that is pharmacy. The whole approach should be, how can we improve patient care? So for that, we have to understand what is happening with the structure, the function, what is the perfect pharmacological approach, what are the adverse drug reactions. So that's the big concept always that has to be in our mind. So now the whole concept starts from neurodegeneration. So what is neurodegeneration? Two important words, irreversible, progressive. So what do you mean by irreversible? Yes, we can't stop the progression of the disease. Once there is an onset, you can't stop it. So what is the therapeutic approach? The only therapeutic approach is to retard, slow down the progressiveness with regard to neurodegeneration. So what do you mean by that? So when there's an onset of disease, the neurons in the cell nervous system or in the peripheral nervous system, they start to function less, or that the neuron starts to die, or there's a reduction in the function in the number of neurons. Okay, so if there is 20 neurons today, tomorrow there's going to be 18, then 16, followed by, so there's a progressive neuronal loss. So the whole therapeutic approach at this point of time is only to retard or slow down the progression of the disease. So that's the concept that I want you to make sure that we understand. We cannot, we are talking about stem cells, we are talking about other surgical approach, but still at this point of time, we only can reduce the rate of neurodegeneration. So the most common neurodegenerative diseases are Alzheimer's and Parkinson's. Okay. So now I just want you to understand that. So what is neurodegeneration? Irreversible, progressive. And the third concept I want you to understand is, with regard to a pathological condition, it is affecting a specific region of the brain. So three things please try to understand. If you're a student, whenever you think about neurodegenerative disorder, number one, irreversible. Number two, progressive. Number three, it occurs in one or a very specific region of the brain. So in my next slide, I'm going to compare Alzheimer's and Parkinson's. A big picture, okay? Very quickly. What is common between Alzheimer's and Parkinson's? Both of them are neurodegenerative disease. What is the difference? One is a movement disorder, other one is a memory disorder. So what is the reason for being one as a movement disorder and the other one being a memory disorder? Simple. Parkinson's disease, as I told you, the region of the brain affected is the nigrostriatal tract, specifically the substantia nigra. So when you have neurodegeneration in substantia nigra, you have movement disorder, which means the nigrostriatal tract is controlling the movement. Very good. Where is the neurodegeneration occurring in Alzheimer's disease? It's occurring in hippocampus and cortex. Hippocampus is associated with short-term memory, and cortex is associated with long-term memory. So if you think about that concept, bigger concept, so what is Alzheimer's disease? It's a memory deficient disorder. So definitely we come to the point, definitely we understand both of them are irreversible, progressive neurodegeneration, but different brain regions. Very good. So with regard to therapeutic approach, whatever we have therapy is just only for the symptom. The current therapeutic approach has nothing to do to reverse the neurodegeneration or to retard the neurodegeneration. So two different words, retard, reverse. So now, so what is the logic or what is the concept behind the pharmacological therapy? It depends upon the neurotransmitters. So at this point of time, I would like you to understand what is neurotransmitter. I'm going to go through that quickly for you. But here, with regard to Parkinson's disease, it's mainly associated with dopamine and acetylcholine. Whereas in Alzheimer's disease, the neurotransmitter affected is acetylcholine and glutamate. So now, if you can understand the difference of the neurotransmission, the therapy becomes very simple. In Parkinson's disease, you have dopaminergic neurodegeneration. What do you mean by that? That is decreased dopaminergic neurotransmission. So what is the therapy? It becomes a simple math. If you have decreased dopaminergic neurotransmission, with regard to pharmacological approach, what do you want to do? You want to increase the dopaminergic neurotransmission. So with regard to Alzheimer's disease, what's happening? You have decreased cholinergic neurotransmission. So what do you want to do? You want to increase the cholinergic neurotransmission. So it's a simple mathematical approach when it comes to pharmacology. So at this point of time, we will stick to that particular point. So we both agree, we all agree that these are neurodegenerative disorders. Very good. So now let's start with Parkinson's disease. Whenever the word Parkinson's disease comes to your mind, immediately, as from a healthcare point of view, it's a movement disorder. There are different types of movement disorder. Okay, these can be associated based on the symptoms. You can have hypokinesia or hyperkinesia. Hypo means less, kinetic means movement, less movement, or hyperkinesia. So with regard to Parkinson's disease, you see akinesia and rigidity. Okay, the muscles are very stiff. So the patient have bradykinesia. Brady means slow. Okay, rigidity. The muscles are so strong. They can't bend. They can't lift something. What does hyperkinesia? You have excessive movement. So the important things you can see is chorea. What do you mean by chorea? The word chorea is associated with choreography. Choreography means dance. So the patient exhibits dance like
selective speaker. Speaker same as system.
Again, it's a hyperkinesia.
Speaker same as system.
We have other different types of movements. But try to understand that you can have the movement disorder classified based on the neuroanatomy too, pyramidal tract. Okay, so with regard to Parkinson's disease, it's an extra pyramidal tract disorder. Okay, number one. Number two, with regard to the symptoms associated with Parkinson's disease, akinesia and rigidity are the most important symptoms. Okay, try to get the point here. Akinesia and rigidity are the most important movement disorder associated with Parkinson's disease. Tremor. Not all patients have tremor. So try to understand the concept. Because of dopamine depletion or dopaminergic neurotransmission in the nigrostriatal tract, the patient usually has akinesia and rigidity. Tremor may or may not be there. So that is the reason you have to understand. The tremor part of the Parkinson's disease is associated with the acetylcholine. So if a Parkinson's disease patient has a tremor, you treat them with a muscarinic antagonist. Okay, atropine or atropine derivatives are used for that treatment. Whereas for hypokinesia, you use drugs that increase the dopaminergic neurotransmission. So that's a very important aspect for you to get from here. So when you think about Parkinson's disease, the first neurotransmitter that has to come to your mind is dopamine. Again, second, you have decreased dopaminergic neurotransmission in the nigrostriatal tract. Other than that, other neurotransmitters that are associated with movement are acetylcholine, serotonin, GABA, glutamate. In Parkinson's disease, the big belief is you have decreased dopaminergic neurotransmission and increased cholinergic neurotransmission, which is associated with a tremor, particularly with regard to tremor. So when you think about dopamine, what should it come to your mind? It's a neurotransmitter associated with love, affection. When a baby is crying, what does a mother do? Or what do? You lift the baby, you hug the baby, you hold the baby. What happens? There is increased amount of dopamine and norepinephrine or endorphins in the brain. So that calms you. When somebody is happy, what do? You hug each other. Why is that? Because that makes dopamine levels go up. Okay? So dopamine is associated with love. I'll give you some example. When you think about depression, what is happening to the monoamine? Or what is happening in Parkinson's disease? You have decreased amount of dopamine. You have decreased amount of dopamine. When you think about what is happening when you have increased amount of dopamine, I don't know how many of you know about Tiger Woods, the greatest golf player. What happened to Tiger Woods? He had so many girlfriends. Okay, I just brought an Indian example to you. When you think about Kamalas and what do we refer to? Kadal Mannan, right? So what is the concept there? Too much of dopamine. Okay, so dopamine is a neurotransmitter that is associated with love. When you think about schizophrenia, I'm bringing a pathological condition here. What is the first pharmacological approach we use in the treatment of psychosis or schizophrenia? It's dopamine antagonist, or we call that as antipsychotic drugs. So the concept is dopamine is associated with movement, mem, and mood. So when it comes to control, start from there. So let's go into a neuroanatomy. So the control, it starts from cortex. From cortex, it goes through the basal ganglia. That's the region in the brain. You can see that I have basal ganglia for you. Then from basal ganglia, it comes to the brainstem. From brainstem, so the brainstem is nothing but the continuation of the spinal cord. From the spinal cord, so now from the cell nervous system, the brain, it goes to the spinal cord, which is the peripheral nervous system. In the peripheral nervous system, you have various nerves. So I have to make sure that you understand that. Now you're talking about autonomic nervous system and you have somatic nervous system. So you have the voluntary control and the involuntary control. So through the peripheral nerves, you have the control of the muscles. So when we think about muscle, immediately you guys, as in the healthcare profession, you should understand there are three types of muscle: the skeletal muscle, the smooth muscle, and the cardiac muscle. So now with regard to the movement, we want to understand the role of skeletal and the smooth muscle. Okay, so try to get the point. So from cortex to the basal ganglia, a region in the brain, usually it's in the midbrain. Cortex, it's the bark. It refers from the word bark, from the outer layer of the tree. So from the cortex, the neurotransmission is passed through the basal ganglia, through the brainstem and the spinal cord, and then it comes to the spinal cord, where in the spinal cord you have the peripheral nervous system, which innervates the muscle, and that controls the movement of the muscle. So that is the most important thing for us to understand. So good. Where did the word Parkinson's disease come from? Dr. Parkinson, a British doctor, a physician. He was the first one to write a particular essay known as shaking palsy. He found that certain patients had tremor. But later on, it was Dr. Charcot, a French scientist, and Tetyakov. These are the people who coined the word Parkinson's disease, and they found out that this particular disease is occurring because of decrease in dopaminergic neurotransmission. So till 1960s, late 60s, we didn't understand what is Parkinson's disease really about. Some celebrities who have Parkinson's disease, who have or had, Muhammad Ali, Hitler, Pope, and recently it's Michael J. Fox, an eminent actor in the US. These people have or had Parkinson's disease. So now, to correlate the current knowledge that we have on Parkinson's disease with the Ayurvedic approach with regard to Parkinson's disease, I want you to understand what is the anatomical aspect of Parkinson's disease, what is the neurotransmitter factor, what are the various biochemical features associated with Parkinson's disease, and how does this neurodegeneration occur, or what is the cause of neurotoxicity with regard to any neurodegenerative disease. So that is the concept we want you to understand. So number one, try to understand that there are very specific tracts in the brain. very specific. Okay, it starts from one place and it goes to another place. So you have major pathways and minor pathways. So when you think about the major pathways, you have the nigrostriatal pathway, you have the mesolimbic, mesocortical, and tuberoinfundibulum. These are the major neuronal tract, dopaminergic neuronal tract in the brain. So here we are going to focus on the nigrostriatal tract. That is the tract that is associated with the control of movement, and that is the tract that is associated with Parkinson's disease. So what do you mean by nigrostriatal tract? You can see it from the brain. It starts, this particular pathway starts in substantia nigra and it goes to nucleus cordalis putamen. Think about that, guys. It's like a map. You start from Chidambaram. How do you want to go to Sirhal? Think about that. Like that, we have a very specific road or very specific tracts in the brain. So here, this particular tract starts in substantia nigra. You can see that in the midbrain region, and it goes to the cordalis putamen. So that is the reason it's called as nigrostriatal tract. What is the function of this particular tract? This particular tract controls movement. This particular tract controls movement in the body. Good. So now, what does neurotransmission? What do you mean by neurotransmission? A neurotransmission refers to neurotransmitter has to be synthesized. For synthesis of the neurotransmitter, you require precursor, enzymes, and cofactors. Once the neurotransmitter is being synthesized, it has to be stored in the vesicles. For the storage into the vesicle, you require transporters, vesicular monoamine transporters. Once it is stored in the vesicle, when you have a calcium influx, the neurotransmitters are being released. Once the neurotransmitters are released, it binds to the postsynaptic receptor for a neurotransmission to occur. So in our case, it's dopamine is being synthesized. It goes and binds to the D2 receptor to control the fine tuning of the body movement. So that is the tract that I want you to understand. If it is a nigrostriatal tract, it starts in substantia nigra, it ends in striatum, but you have dopamine being synthesized, stored, released, binds to D2 receptor, where you control the movement in the body. Very good. So to follow that up, how does the neurotransmission is being terminated? There are three processes. One, you have enzymes that degrades the neurotransmitter. Number two, you have reuptake. And number three, you have autoreceptors. So stimulation of the autoreceptors decreases the neurotransmission. So this basic concept for every neurological disease, we have to understand that. If you understand the neurological process, the neurotransmission, you can tackle any pathological condition. Okay, good. So now with regard to dopamine synthesis, you know that it all starts with L-DOPA. There's a reason I'm going through this, because when you come to mucuna pruriens, I want you to make sure you understand how dopaminergic neurotransmission is being affected in Parkinson's disease and how mucuna pruriens and Ayurvedic botanical can overcome these problems. So when you want to think about dopamine synthesis, it starts from tyrosine. Tyrosine is being converted to levodopa. That's the rate limiting step. So you require a precursor, that is tyrosine. Then you have the tyrosine hydroxylase enzyme. You have tetrahydrobiopterin, which is the cofactor. Levodopa is being formed. Levodopa in the presence of aromatic amino acid and decarboxylase enzyme and pyridoxal phosphate cofactor, dopamine is being formed. So this is the basic concept. Why are we talking about that? Because when you think about Parkinson's disease, you have dopamine being depleted in the striatum. So now what can you do? How can you increase the synthesis of dopamine? So we can use levodopa. That's the first concept I want you to translate, how levodopa is connected to Parkinson's disease treatment. So again, try to get the point here. Synthesis, substantia nigra. So dopamine synthesis starts there. It is being released into the synapse. What is a synapse? Synapse is the region between two neurons, It's the space. It's the gap between the two neurons. So dopamine binds, and then you have a neurotransmission. And dopamine is being metabolized by two enzymes, monoamine oxidase B and catecholamine O-methyltransferase. So you can see that from the cortex through the basal ganglia, it goes to the spinal cord and it controls the muscle activity. When you have loss of dopamine, the problem is you also have a disturbance in acetylcholine. So what is happening? You have increased muscle tension, which leads to rigidity, which leads to hypokinesia. Sometimes it leads to tremor. So it's a very simple concept. When you have lack of dopamine, you have increased acetylcholine. That causes enhanced muscle contraction, which leads to rigidity in the Parkinson's disease patient, which leads to hypokinesia, because when the muscles are rigid, they can't easily move. They can't lift something. Same time, it also can result in tremor. It also can result in tremor. So the most important concept when it comes to Parkinson's disease is loss of dopaminergic neuron. And then you have certain protein getting accumulated, like alpha-synuclein, okay, Lewy body. Again, you can very well see that the problem is neurodegeneration in substantia nigra. You can see that neuronal loss in substantia nigra, which leads to striatal dopamine depletion. Again, you can see that tyrosine hydroxylase expression is decreased. What do you mean by that? Tyrosine hydroxylase is the rate limiting enzyme in the synthesis of dopamine. You have less tyrosine hydroxylase. That can lead to less dopamine. Altered glial activity. What do you mean by that? When somebody talks to you about brain, there are two different types of cells in the brain. One is neuron, the other type is glial cells. Glial cells are supporting cells. They support the neuron. They maintain the neuronal health. They maintain the neuronal function. When you have altered glial activity, it can result in increased oxidative stress and inflammation. Good. Okay. Other thing is, as I told you, you have Lewy body accumulation and increased iron content. So again, I want you to understand in Parkinson's disease, increased iron content can be attributed towards dopaminergic neurodegeneration. Very good. Okay. So we talked about the biochemical imbalance. You have less dopamine and increased cholinergic neurotransmission in Parkinson's disease. We understand the concept there. So now, this is a very important concept to understand. Whenever you think about neurodegeneration, how does neuronal death occur? There are 5 common mechanisms. Number 1, oxidative stress. What is oxidative stress? Where you have less antioxidant and more pro-oxidant that causes oxidative stress. Same concept with inflammation. More pro-inflammatory mediators and less anti-inflammatory mediators that leads to inflammation. What is apoptosis? Programmed cell death. The cell gets ready to die. Excitotoxicity. It means glutamate acts on the glutamate receptor. It increases the calcium influx inside the cell, which causes cytotoxicity or neurotoxicity. What is mitochondria? Mitochondria is the powerhouse of the cell. Whatever we consume, whatever we eat, has to be converted to ATP for the cells to function very well. So mitochondrial dysfunction. So these are the 5 major concepts of neurodegeneration or neurotoxicity: oxidative stress, apoptosis, inflammation, excitotoxicity, and mitochondrial dysfunction. These are the major concepts associated with cell death. Very good. So now, what are the current therapeutic approach for Parkinson's disease? As we told that, number 1 approach is to increase the dopaminergic neurotransmission. So we can use the precursor for dopamine, levodopa. Levodopa cannot be given alone, so it has to be given with a peripheral amino acid decarboxylase inhibitor, carbidopa. Number 2, you can give amantadine. I think this particular drug blocks the reuptake and increases the release. Number 3, dopamine agonist, specifically D2 agonist. So you increase the synthesis, increase the release, stimulate the postsynaptic receptor. That's one concept. The other concept is block the breakdown of dopamine. So what are the 2 enzymes that is associated with the dopamine breakdown or metabolism? That is monoamine oxidase B and COMT, catechol-O-methyltransferase. So we use MAO inhibitors and COMT inhibitors. There are very few neuroprotectants that are being used. That is the whole problem with this particular pharmacological approach. We know that it's a neurodegenerative disease. We definitely know that there is dopaminergic neurodegeneration occurring, but still the current therapeutic aspect only focuses on the symptoms. We are only looking into the treatment of the hypokinesia aspect or the tremor aspect associated with Parkinson's. So that's a major problem that we are facing with regard to the therapeutic approach. Okay. So now what are we doing? What are we trying to understand? Can we block oxidative stress? Can we block apoptosis, reduce inflammation, increase the mitochondrial function? So what? Thereby, can we retard the neurodegeneration? So try to understand the concept. Ayurvedic approach is not a symptomatic approach. We are trying to understand how we can reduce the neurodegeneration associated with the neurotoxicity. That's the whole concept. So to do that, we started with Mucuna pruriens. Okay, so we want to have an in vivo model where we can test Mucuna pruriens, or we can validate the use of Mucuna pruriens in Parkinson's disease. We not only did that, we we compared mucuna pruriens effect with the standard. What is the gold standard currently in the treatment of Parkinson's disease? Levodopa. So we compared the effect of mucuna pruriens with levodopa. So that was the first thing. So we initially started with mucuna pruriens. We got it from Zandu. Why did we want to buy it from Zandu? We want to validate the product. Zandu controls the growth of the botanical. They standardize the botanical. They have the aspect how much amino alkaloids are there. They know how much other components are. So we got from Zandu Pharmaceutical. So mucuna is from the Leguminaceae family. So for the first study, we want to see the chronic effect of mucuna pruriens extract on monoamines. So we took rats. We administered mucuna pruriens various doses, 2.5, 5, and 10 grams. We mixed it with the feed and we administered for 52 weeks. That's for 1 year. It was a lot of money was involved. After that, we collected different regions of the brain. We took the nigrostriatal tract, substantia nigra striatum, good. We also took hippocampus and cortex, and we also took out blood, good. So now, we analyzed, the first study, we analyzed the amount of levodopa. Why did we go for levodopa? So, we all know that historically, people with Parkinson's disease were treated with mucuna. Okay, for centuries in Ayurveda, we have reports, we have well-documented literature to prove mucuna was used. So now what is the concept we are trying to bring is, okay, mucuna contains levodopa. So as a positive marker to support our study, we measure levodopa in the blood. So as compared to the control, mucuna administration, okay, where we had levodopa alone, mucuna had higher amount of levodopa in the blood as compared to levodopa alone. Good. So that was by administering an endogenous compound which contains levodopa, but it exhibited higher amount of levodopa in the blood. That's a great study. Second study, in the brain. So first question is, neuroanatomically, where should we have high amount of dopamine? It is in the striatum. We proved that. So if you see the graph, highest amount of dopamine was present in the striatum. So we saw a steady state when we administered 2.5, 5, and 10 gram per kilogram. It's a lot of mucuna pruriens. When it was mixed with the food, this is rat, they eat a lot. Okay, you have to understand that the amount of dopamine was higher. Very good. Okay, good. That's the first study. And then that study also proved that there was no toxicity associated with the administration of mucuna. That's very important. So whenever we administer as a pharmacist, it's not always about pharmacology. We have to always think about hypersensitive reaction and adverse drug reaction. For a year, we didn't lose any animal when we treated them with mucuna pruriens. Then we want to make sure that we can validate it using HPLC and spectrophotometric analysis. What compounds can we think? So definitely levodopa comes to our mind. Yes, we did that. But not only that, we also found out NADH and coenzyme Q10. So if you're a pharmacist, you definitely have to know about coenzyme Q10. It's a very important component of the mitochondria. Yes, there are a lot of formulations currently available which contains coenzyme Q10, and it is being given for the welfare. Okay, it's kind of like a vitamin. It's a prophylactic drug which can be used to decrease neurodegeneration or specifically cardio problem. Okay, coenzyme Q10 is a great drug with very minimal adverse drug reaction, but it has a protective effect. It's very similar to your vitamins, multivitamins. Okay, we validated them, and we were first to show that coenzyme Q10 and NADH are present in mucuna pruriens. Okay, again validating it. That coenzyme Q10 has a lot of literature are currently available that shows that coenzyme Q10 exhibits neuroprotection, cardiac protection. Good enough. Now, the second study. Okay, first we have proved that yes, mucuna pruriens increases the levodopa concentration. Mucuna pruriens is non-toxic. Very good. So in the second animal model, what did we do? We want to generate an animal model that is a valid animal model for Parkinson's disease. So now we took a rat. Okay, good. We administered 6-hydroxydopamine. So I'm going to use what is 6-hydroxydopamine? It's a neurotoxin. So you can see that. So we administered this particular drug in the intranigrally. What do you mean by that? You cannot administer the drug peritoneally. Why? Because it cannot cross the blood-brain barrier. So we have to administer the drug intranigrally. That's 6-hydroxydopamine. Very good. We waited for 4 weeks. Okay. Now, let me tell you that we only administered the 6-hydroxydopamine in one side of the brain. We only administered in the 6-hydroxydopamine in one side of the brain. We call that unilateral administration. Unilateral administration. So what happens during the 4 weeks? Because of the toxicity, in one side of the brain, you have less dopamine. In the other side of the brain, you have more dopamine. That's the dopamine imbalance in the brain. If I administer on the right side, after 4 weeks, I have less dopamine on the right side as compared to the left side. Okay, that's a dopamine imbalance. Now, that is the beauty of pharmacology. If I give a drug that's a dopamine agonist, what do you mean by dopamine agonist? If the drug binds to the dopamine receptor, the animal rotates contralaterally. What do you mean by contralaterally? If the lesion was done, if the surgery was done on the right side, the animal starts rotating in the opposite direction. Okay. Whereas if I give amphetamine, a drug that increases the release of dopamine, the animal rotates ipsilaterally. So that is a very beautiful experiment for us to understand. So now when we administer 6-hydroxydopamine, the 6-hydroxydopamine lesion rat, when we administer mucuna, the animal rotated contralaterally. So that was a great finding for us, which means mucuna pruriens is not only levodopa, it has other components that makes the animal to rotate in the contralateral rotation, which means mucuna pruriens exhibited a dopaminergic agonist effect. Very good. That was the first thing. Okay, yes. Now, can it exert? That is a behavioral experiment. You can see that. This is a rotor rod. When you administer that, our animals rotated contralaterally. And we use amphetamine as a positive control. In pharmacology, positive controls are very important to prove your pharmacological concept. So when we administer amphetamine, the animals rotated ipsilaterally, same side towards the lesion. Whereas when we administer mucuna pruriens, the animals started to rotate in the opposite direction. That is in the contralateral direction. So not only that, when we measured the dopamine and norepinephrine level in the HPLC, we saw that mucuna pruriens protected or blocked the 6-hydroxydopamine-induced dopamine depletion. That's a very important thing. So we had neuroprotective effect of mucuna pruriens. Very good. So now, what is the reason? How does this happen? Because number one, mucuna pruriens, as I told you, it has NADH, it has coenzyme Q10. So these can be associated with the mitochondrial function. So mucuna pruriens dose-dependently increased the complex 1 activity in the mitochondria. So as I told you, mitochondria, you have various electron transport chain that transfers the electron to produce ATP. So when you can increase the complex 1 activity, mucuna pruriens can increase the mitochondrial function, thereby it can reduce neurodegeneration. First concept. Second concept, we talked about apoptosis. Apoptosis is nothing but programmed cell death. Okay, so now what happens in programmed cell death? We have a lot of caspases getting activated. Those are enzymes that results in DNA degradation. Mucuna pruriens. So we took plasmid DNA and we took genomic DNA, and we studied the neuroprotective effect of mucuna. Mucuna blocked, okay, the levodopa induced. So levodopa induced genomic and plasmid cleavage. So now think about that. We are talking about number one, or the gold standard drug in the treatment of Parkinson's disease. What does this do? This particular drug can cause DNA damage, whereas mucuna blocked the DNA damage that was done by levodopa and copper. So that's another thing. So it exhibited, number one, increased mitochondrial function. Number two, it can block apoptosis. Number three, we have published where we show that mucuna exhibits antioxidant and metal chelating activity. Why am I talking about metal chelating activity? We clearly told you that there is increased amount of iron present in the brain of Parkinson's disease. Mucuna pruriens can chelate. We know that from pharmacologically. They can form a complex and they can remove the toxicity of the iron. So it can have a chelating potential. That's what we published. Number two, it can block the generation of the reactive oxygen species. Reactive oxygen species are directly associated with oxidative stress. What happens when you have increased reactive oxygen species? It can cause peroxidation of the lipid. Lipids are present in the outer cell membrane. That causes neurodegeneration. So now we are clear. So definitely, mucuna pruriens increases the mitochondrial function. Number two, it blocks oxidative stress. It reduces apoptosis. Thereby, definitely we know that it can reduce neurotoxicity. That's important. So one more thing before I find out, one of our finding again with regard to Parkinson's disease and with regard to Mucuna is try to understand that there are different types of dosas. Okay, this is based on the body mass index, Vata, Pitta, Kapha. So the most concept that one of the concepts we found out also was Parkinson's patients are mostly Vata type. Their BMI is less than 25. Okay, so the patients with decreased BMI, they are prone to neurological disease, and Parkinson's disease is a Vata-based disease. So for an example, when you think about the Kapha, where you have the BMI more than 30, it's usually associated with diabetes mellitus, endocrine disorders, or bone diseases. That makes sense. Okay, so that is with regard to Ayurveda and dosas. So now, how did we validate Mucuna pruriens for movement disorder? Yes, it blocks dopamine depletion. Very good. Okay, neuroprotective effects are seen in the nigrostriatal tract because the animal can rotate in the contralateral side behaviorally. Number 3, what are the possible mechanisms of neuroprotection? It reduces reactive oxygen species, which means reduces oxidative stress. Because of decreased oxidative species, it can prevent lipid peroxidation. Great. Number 3, increases the mitochondrial complex 1 activity, which means it's going to increase ATP content and decrease the mitochondrial dysfunction. Blocks DNA damage, thereby it can protect against apoptosis or programmed cell death. Definitely, we are sure, we have published this, so I can say that Mucuna pruriens exhibited a higher neuroprotective effect as compared to the gold standard in the treatment of Parkinson's disease, which is levodopa. So definitely, I feel very happy that we can say with regard to movement disorder, Mucuna pruriens has been validated to prove a higher neuroprotective effect. Definitely, that concept we have proved. Very good. Now, let's go quickly to the memory aspect, that is dementia. So think about dementia. It is not a specific disease. It's a group of symptomatic pathologies where people have difficulty in memory, thinking, social ability. They can't behave properly. Okay, they cannot. When you have the problem with dementia, a patient cannot function with their daily activities in a proper manner. That's the concept. Okay, there are different types of dementia. The most common type of dementia is Alzheimer's disease. Okay? When you think about that, Alzheimer's is one of the most common type of dementia. So when you think about dementia, yes, it's a memory impairment. How does it differ from normal aging? Always ask that question. Yeah, during the process of aging, we have cognitive impairment. Okay, so in both, you have to understand that they both forget an experience. But in Alzheimer's, they forget the entire experience. In normal age-related disorder, you only forget about a little thing. But here, they forget. A normal aging adult, they can follow instruction. Whereas an Alzheimer's patient, they cannot follow any instruction. You have to take care of them as if they are a newborn baby. Okay? Unable to care for them, a dementic patient, an Alzheimer's patient cannot care for themselves. So it's going to cost billions and billions of rupees or dollars to take care of these patients. So think about that again. Whenever you think about the neuroanatomy, think about the neuron, think about the glial cells, and think about the cerebrospinal fluid. You can see that is a nice cartoon to see for you. You have think about the neurons, you have the glial cells and the CSF. So again, when you think about Alzheimer's disease, 2 major regions. Okay, hippocampus is associated with short-term memory, and prefrontal cortex is associated with the long-term memory. Okay, think about that. Hippocampus, short-term memory, and cortex, prefrontal cortex, specifically associated with the long-term memory. So again, Alzheimer's, like Parkinson, is a irreversible and progressive deterioration of the neurons, which is associated with intellectual capability and personality. So that's a personality change, and their complete intellectual capability is completely destroyed. It's completely destroyed. So with regard to Alzheimer's, always think that we know they have cognitive impairment. If you are a student, try to understand, guys, they also have severe neuropsychiatric changes. They have psychotic phase, they have depression. Okay, so they undergo, they have anxiety, they go, a variety of neuropsychiatric changes can be seen with Alzheimer's patient before they have cognitive impairment. Okay, so now again understand the concept. So like for Parkinson's disease, it was the nigrostriatal tract. Whereas when you come to the cholinergic pathway, here it starts with basalis minor. This particular cartoon, you can see that. From here, the cholinergic neurons projects to the cortex and hippocampus. So with regard to the cholinergic pathway, you have neurodegeneration in cortex and hippocampus. That's the reason you have a deficiency in the short-term and long-term memory. So with regard to neuropathology, what is happening in Alzheimer's brain? Number one, you have accumulation of two important proteins. One is beta amyloid, the second one is tau protein. You have two pathologies, tau protein accumulation and beta amyloid accumulation. So beta amyloid is a protein which is formed from amyloid beta protein, precursor amyloid beta protein. There are two enzymes, alpha beta secretase, which is associated with the degradation. There are secretases, there are three secretases, alpha, beta, and gamma secretase. Two secretases are associated with the formation of beta amyloid, which is toxic. There's one secretase which is associated with the breakdown of the alpha, the beta amyloid. So secretase is an enzyme that helps the conversion of the precursor amyloid beta protein to beta amyloid, either 40, 42. There are numerous, okay, beta amyloid, but 40 and 42 have been well established to provide neurotoxicity with regard to Alzheimer's disease. So now, how does beta amyloid induce toxicity? Okay, so they can act on nicotinic receptor. Okay, they can act on NMDA and AMPA receptors. So which means beta amyloid can affect the cholinergic neurotransmission, and beta amyloid can affect the glutamatergic neurotransmission. Okay, that's the big picture. At least try to understand that. Why am I going with that particular thing? Because as I told you, for Alzheimer's disease, currently we have two pharmacological class of drugs. Number one, cholinesterase inhibitor. What do you mean by that? A drug that increases the cholinergic neurotransmission by preventing the degradation of acetylcholine. Very good. So we have donepezil, rivastigmine, galantamine. With regard to NMDA receptor, again, if you try to understand, NMDA receptor is associated with excitotoxicity. What do you mean by excitotoxicity? Okay, when glutamate binds to the NMDA receptor, it causes calcium influx, which causes the neurodegeneration or neuronal cell death. So you want to prevent that. So we use NMDA antagonists. We have donepezil, rivastigmine, and galantamine, which are cholinesterase inhibitor, which are reversible or semi-reversible cholinesterase inhibitor. Very good. So with regard to Alzheimer's disease, you want to approach a multi-targeted ligand, not only for Alzheimer's, Parkinson's too. So try to understand that we want to make sure that we not only approach on one target, not only on cholinesterase inhibitor, not only on NMDA. Let's see whether we can block the production of beta amyloid. Can we inhibit secretase? Can we reduce inflammation? That is the multi-targeted ligand approach. Okay, so that's what we want to take into our account. So whenever we think about that, first we want to establish the in vitro neuroprotective effect of Centella asiatica. So we pick Centella asiatica and Ayurvedic botanical. Okay, we want to see whether Centella can enhance the synaptic function, strengthen the synapse so that the neurons can communicate properly, which can reduce neurodegeneration. Okay, and then we want to go to the in vivo effect. So with regard to Centella asiatica, we all know that, okay, it has asiaticocytes, asiatica, brahmocytes. Okay, that is Centella asiatica. It has been used to treat different diseases, variety of diseases. Centella asiatica, from cancer to peripheral disorders to neurological disorders. Okay, literature has shown that. So initially, we want to see whether it can reduce inflammation, whether it has antioxidant activity or anti-apoptotic activity. So we initially evaluated with H19 hippocampal neurons. So that is the beauty when you do an in vitro experiment. We can have the specific neuron. So we want to take the hippocampal neuron. And then we also looked into what is happening with the glutamatergic receptor, and does this extract, Centella asiatica extract, does it have neuroprotection? Okay, so we studied few studies there. So definitely we validated Centella asiatica. Definitely it had polyphenols, it had flavonoids. Okay, so definitely other than the alkaloids, it exhibited flavonoids and polyphenols. Very good. So when it comes to our salient findings with regard to the in vitro study, Centella had polyphenoids and flavonoids. It inhibited the cyclooxygenase and lipoxygenase activity. So now try to understand, you are blocking the pro-inflammatory mediators, which means they exhibited potent anti-inflammatory effect. Very good. Number two, it inhibited caspase 3. Caspase 3 is the most important enzyme that is associated with the cleavage of DNA. which means it can reduce apoptosis. This is an in vitro study, guys. This was done, okay, in vitro study. It can reduce the formation of the pro-inflammatory cytokine TNF-alpha. Very good. Then, with regard to our neuronal effect, it can modulate the NMDA receptor, which means it can reduce excitotoxicity. It will modulate SCAM2 and CREB. These two proteins are associated with synaptic function, which is associated with the neuroprotection. It increases BDNF, brain-derived neurofactor. What do you mean by that? It makes the neurons more viable or reduce the neurodegeneration. So the big picture is it can decrease excitatory neurotoxicity. Not only that, we challenged or we studied, we investigated the neuroprotective effect of Centella against hydrogen peroxide-induced neurotoxicity. Hydrogen peroxide is an endogenous neurotoxin. It's being generated in the brain. So we definitely saw that Centella can survive against the hydrogen peroxide challenge by decreasing the reactive oxygen species, which is reduced oxidative stress, and decreasing excitotoxicity. So with regard to excitotoxicity, we saw that one of the neurotransmitters, NR2B receptor, which is associated with excitation. So we saw the NR2B expression to be decreased, and we saw NR2A expression to be increased. Amazing finding. Okay. Now, try to get the point. So now we have proved it in vitro, okay, in a cell outside the body. Now we want to see it in the in vivo. So when we thought about in vivo, the first thing is we need an animal model. So for this particular study, we used a double transgenic mice. So this particular mice had two genes, extra genes, amyloid precursor protein and presenilin gene. So what happens when you have these two genes? You are going to have increased beta amyloid production in the brain that causes cognitive impairment. Very good. Okay, so you can see that beta amyloid content and secretase activity was increased in the transgenic mice. We have published this publication. Okay, so you can see that in the cortex and hippocampus, we saw increased beta amyloid, and that's because of increased secretase activity. So definitely we are now understanding that when you have increased beta secretase, you have more beta amyloid formation. Very good. Okay, then we saw increased markers of oxidative stress. Definitely we saw glutathionein peroxidase, reactive oxygen species, lipid peroxidation went up. Okay, and we also saw increased COX activity that's associated with inflammation. Okay, pro-inflammatory cytokines. Okay, TNF-alpha was increased. You saw IL-1 beta was increased. So definitely in the brain of these Alzheimer's disease model, that is the trouble transgenic mice, we saw increased pro-inflammatory cytokines. So what when you have increased pro-inflammatory cytokines? Okay, you have that affects the neuronal firing. So we studied based on the excitotoxicity, we studied on that, and we found that definitely we have increased NR1, we saw decreased PCREB, decreased kinase. All these is also attributed to the decreased, okay, neuroprotective effect. So that is the main thing that I'm talking about. So now let's validate our animal. So now we have proved, okay, increased secretase. So what? More beta amyloid. So what? More inflammation, more neurotoxicity. How can you prove that? We did two. We did biochem, we did behavioral experiment. We did open field and Y-maze. So with regard to open field and Y-maze, they can talk about the anxiety of the animals, and they talk about the memory of the animals. So these two techniques were used, open field technology and Y-maze, and we clearly found that the animals had behavioral deficiency. The animals were more anxious. The memory impairment has set in these animals. So definitely now we have validated our animal model. How did we validate? Biochemical, neurochemical. We saw neurotoxic markers increase, and that caused behavioral deficiency as seen in open field and Y-maze. Very good. So now you can see that we saw behavioral deficits, we saw excitotoxicity, we saw inflammation, and we also saw deficits in synaptic plasticity. Very good. So now we have a valid animal. Okay, we have a valid animal to study the neuroprotective effect. You can see that. You can see that very well. You can see that definitely we saw inflammation, mitochondrial dysfunction, reactive oxygen species that caused behavior. An animal model. So we want to study the effect of Centella asiatica. So we mixed with the feed. We had two doses. Okay, we had a non-transgenic group, we had a transgenic animal group, and we have 2 doses of Centella asiatica administered to the transgenic animals. So number one, the body weight and the behavioral changes were same. What do you mean by that? The animals were eating normally because these Ayurvedic materials were combined or mixed with the feed and administered to the animals. So definitely we want to make sure that we had to watch the body weight. And now the body, based on the body weight, we definitely validated that the animals were eating properly. The body weight changes were comparable to the control, a non-treated drug control group. Good. And then number one, behavioral changes. Yes, Alzheimer's disease model, APP mice, showed decreased function in the Y-maze test, and definitely central treatment improved the behavioral deficiency. That was a very good finding that we had. Okay, so behaviorally, we proved that central can overcome the behavioral deficit induced by the animal model. So what is the next marker? Very important. The most important marker is beta amyloid deposition. So Centella asiatica can decrease the beta amyloid formation that was associated with these particular trouble transmission. That was a great finding. You can see that in the control, we didn't have anything. And then when you administer these to the trouble transitioning mice, you have decreased production of beta amyloid. So that was a very important finding. And we saw that both in the cortex and hippocampus. We saw that both in the cortex and hippocampus, and that can be attributed to the decrease in the beta secretase activity. So that makes logic. You have decreased beta secretase activity that's leading to decreased beta amyloid formation. Good enough. Then very important is TNF alpha. TNF alpha is a pro-inflammatory mediator that has shown to affect the beta amyloid formation. Centella definitely dose dependently decrease the beta amyloid formation, Not only beta amyloid formation, it decreased TNF alpha and interferons alpha. Both the pro-inflammatory mediators were decreased by Centella asiatica. Okay, so we looked into other pro-inflammatory cytokines too. So these are the various other pro-inflammatory cytokines that we had looked into the interleukins, interferons. We looked into several pro-inflammatory mediators. And then we also looked into the T cells. So now the big picture, when you think about Alzheimer's disease, a lot of people think that Alzheimer's disease is a neuroinflammatory disorder, and T cells play a vital role with that. And so now you can see from that picture is Centella therapy can reduce the T cell mediated pro-inflammatory response in the brain. Okay, so as I told you, this was a very big study. We looked into several mediators, several pro-inflammatory mediators in cortex and hippocampus. And the beauty of the study is, that is the beauty of Ayurveda. It's not one ligand-based approach. It's a multiple ligand-based approach. Again, going back to our theory, Centella can attenuate the pro-inflammatory cytokines, reduce inflammation, thereby it can exhibit neuroprotection. So now from the inflammatory pathway, we go to the oxidative stress pathway. Definitely Centella reduces reactive oxygen species. Centella block the lipid peroxidation induced by reactive oxygen species. And then with regard to excitotoxicity, we measured glutamate and GABA level. Definitely Centella decreased the glutamate level, and also it decreased the GABA level, which means it can reduce anxiety. Okay, good enough. And then we did some electrophysiological recording in the brain. That's where we are looking into the modern technology, and we proved with electrophysiology again, Centella can reduce the excitatory neuronal firing. Therefore, it can reduce excitotoxicity. It can reduce the excitotoxicity. To conclude, so definitely we had a valid model. Double transgenic mice, APP, PS, and Y. Centella asiatica can affect the excitotoxicity. It can increase the synaptic function, okay, reduce beta secretase. It can prevent the T cell stimulation mediated pro-inflammatory cytokine response, reduce inflammation, reduce oxidative stress, okay. Thereby, it can provide neuroprotection. Thereby, it is a multi-therapeutic effect, multi-director ligand effect, anti-inflammatory effect, inhibit beta secretase, antioxidant effect, okay. So it exhibited multiple neuroprotective mechanisms. The last question, if you are given a chance, do you want to live with Parkinson's or Alzheimer's? Always, guys, I'm going with Parkinson's because you are dead. You are a living dead person with regard to Alzheimer's. So that's a kind of a clinical question just for us to think about, okay? I would like to thank all my collaborators, all my students who have helped me to be here. Definitely, I owe everything to Annamalai University where I started my career. Okay. I thank Dr. Maniam. He was a physician, a neurologist who trained me in this field of Ayurvedic in America. He's no more. He was a neurologist from India who I worked with. He was the one who patented Mucuna Zandu, first one to do that. Then we carried out the work with Mucuna pruriens, Alzheimer's, and Parkinson's disease. Since Dr. Maniam is no more, thank you, sir. And then, thanks so much, guys. If you have any questions, please let me know. I'm from state of Alabama. I work at Auburn University. I sincerely thank you for this particular opportunity. Thank you, everyone. So, Dhanapal sir, I'm done, sir. Thank you, Dr. Manna. Thank you, Dr. Mohanda.
Thank you, Dr. Murali. Thank you so much. You spent a lot of time. Thank you so much for your detailed presentation.
Thank you, sir.
Venkatesh, Dr. Manna sir is there, no? Dr. Venkatesh, you unmute Dr. Manna sir, please. Venkatesh? Yeah, just a minute, Murali.
Yes, sir.
I have a
Yes, sir. Yes, sir. Yes, sir. Yes, sir. You can proceed, sir. No problem.
Dr. Krishnan.
Yes, sir.
But thing is, Ayurveda is much more broader than simple botanicals or the herbs. In our modern terminology, we have introduced phytopharmaceuticals. In our legal statute, we have introduced phytopharmaceutical. But these are all oriented towards the modern medicine based on the experimental research we do. But Ayurveda, or in that respect, any Indian system of medicine like Siddha, they are altogether little, in my feeling, they are little different.
So what do you mean by different, sir?
Can go ahead, popularizing our Ayurvedic science or the Siddha, how we can do this so that people will accept this as a system.
So as a pharmacist, we have to step in, sir. Number one, adulteration, sir. We have the whole Ayurvedic approach became very low because of adulteration, sir. People became very greedy. They started adulterating the botanicals. That's number one, sir. So if you can have a strict guideline to control the quantity of the product, we are going to excel, sir. That's number one. And then number two, package, sir. How are we going to formulate it? What is the dose? What is the formulation system that we are going to use so that we can be equivalent to the current modern therapeutic? Because we have a superior pharmacological product with us. The problem is we are not controlling the toxicity. We are not controlling the adulteration. Number two, the formulation is not very viable for the patients. We have to make it more attractive. How can we make it more attractive? So the patient can consume these products more. Not only for therapeutic, prophylactically, how can we come? We are better than the multivitamins available in the market. Ayurvedic products are far better because it has been proven to have less toxicity. As I told you, for Mucuna, we did a 1-year study. And we didn't see any hepatological toxicity. We didn't see any renal toxicity. So the problem comes with, that's the reason I told that we got it from Zandu. So Zandu took a lot of time for us to give the botanical to us, because they validated when they were growing, at what season you had different alkaloid content. They validated the botanical. The whole problem is a lot of people are very greedy. They want to make quick money. They forget the logic, the science behind that. So the adulteration sets in, or the proper extraction procedure is not being there. So like what a very valid drug, we also have to have a very strict guideline for our Ayurvedic. That way we can have a very beautiful product, sir.
Thank you, Murli. But
Yes,
you know, when we talk of the adulteration or the contamination or deliberate addition of some allopathic medicines into it, that is what people say. But when we look at the quality of allopathic medicine, whether it is used in India or it is used in America, there are still problems of these impurities remain. Because we have seen in the recent past, this ranitidine with nitrosamine
cancer. Yes, sir.
contamination, we have problem. We have problem now with the metformin.
Yes, sir.
Because so it's not that they just deliberately think that the Ayurvedic products are more with the quality issues.
Yes, sir. So think about that. So if you think about Mucuna pruriens, it contains levodopa. So we are now comparing apple to apple. We are comparing a synthetic levodopa with the levodopa which is present from a botanical. And we have clearly shown that the botanical levodopa has superior neuroprotective effect, or let me not use the word superior, significantly higher neuroprotective effect as compared to the synthetic levodopa, number one. And then the other way around, if you're talking about, let's forget about the impurities. Let's now validate the other nutrient present. So you told a very nice word, phytonutrients. Let's validate that. So a pharmaceutical company, when they are going to sell an Ayurvedic product, they should tell that our alkaloid content is between 20 to 25%. Coenzyme Q10 content is around 2%. NADH content is 5%. Flavonoid is 20%. Thereby, we are validating our product. That's another problem we are not having, because everything we are just telling the word extract, which is a very loosely turned term. But when we say that we had coenzyme Q10 3% in our extraction, we had flavonoids 5%, we had alkaloid content, total alkaloid 20%, protein concentration was 4%. So when you can go from there, polyphenols 5%, I'm just giving you a tough number. That by you have validated the product. Let's forget about the adulteration. But now, like we say that this particular drug has 200 milligram of aspirin. So now we are coming with the Ayurvedic product where we have alkaloid content is 20% in our product. So some other company can say that we have 30% alkaloid. We can say that micronutrients, let me use the other word, micronutrients, coenzyme Q10 is 3%. This is 2%. Thereby we are validating in. So that is where we have to come in now. We have to say that our product is validated. Anytime you take it, you're going to have this amount of micronutrients and this amount of alkaloids in it. Thereby, we are very much strengthening the whole approach, because nobody wants to come and do that with regard to the micronutrients and with regard to the alkaloids or flavonoids. They don't want to come and tell how much percentage. That is the problem we are having, sir.
Thank you, Murli. Dhanapal.
Yes, sir. Yes, sir. Murli, there is a question from the audience. Is there any natural remedies to improve memory power?
Sir, sir, one thing, first to start with this, it's genetics, sir. Alzheimer's disease is mainly seen in Western countries, higher in Western, significantly. I'm using the word again and again, significant higher because of the statistics. Significantly higher in the Western countries as compared to the Asian countries. To start with this, genes. So if you have a good genes, we don't need to worry about memory. With regard to memory, you want to increase there are two approaches here, sir. One, to increase memory, to block the cognitive impairment.
Yes.
So it's a kind of a same coin, but two sides. Am I going to increase the memory or decrease the cognitive impairment? So the problems are the current therapeutic drugs that we have cannot be used, because if you're using it in a normal patient, they have so many adverse effects. See, now the problem is whatever drugs that have been approved for the treatment of Alzheimer's cannot be used for any normal patient because of the adverse effects. With regard to memory enhancer, a lot of companies have come with a lot of products, sir. I strongly believe coenzyme Q10 is a good one because it's like multivitamin. It's going to increase the ATP formation. More ATP, more neuronal function that can enhance memory. So I'm just going with a very basic, it's very superior product as compared to multivitamin, coenzyme Q10.
Yeah. Thank you, Murali. Another one is
Yes, sir.
whether Ayurveda provide treatment for addiction.
Yes, sir. We have, sir. Very nice question, sir. Sir, ganja, currently cannabis is a hard drug, sir. Cannabis is a hard drug that's being used for opioid addiction, sir.
Oh.
Can you believe that, sir?
Yeah.
It has been approved. That was, it has been approved by FDA. Cannabis can be used. In US, they are going. Currently, we have 6 states where you can grow cannabis in your house.
Oh.
You can grow cannabis at your house. If you have a medical condition, you can grow cannabis in your house legally. So now cannabis has been approved for the treatment of opioid addiction. So I answered your question.
Thank you, Murali. Now,
Anytime, sir.
Dr. Manna sir to say a few words. Please, sir. Manna sir.
Good morning, Murali. It was a nice, informative talk, and hope all the participants have benefited, enlightened, and they may be motivating part with the pharmacologist to adhere to this research, this area, and promote our Ayurvedic medicines, natural medicines available in our country. Rich source we have from south to north. We have a rich natural source of the medicinal plants, and hope the pharmacologists, more and more pharmacologists will be working on this and promote our Ayurvedic system of medicines, which is I am very happy the today's speaker on this Ayurvedism, where the Ayurveda is the natural system medicines originated in India 3,000 years back, and the speaker talking to Indians away from India and Indian at home. I hope he feels at home while addressing the people in India. Of course, there are many international participants. It was an excellent talk, very encouraging and informative talks. Thanks, Murali. And Murali, ganja, you say, is a treatment for opioid addictions.
Yes, sir.
Ganja itself is traditionally from very age-old plant used for addiction by Indians.
Yes, sir.
So it itself will cause addiction because ganja is used for long, and it is a raw material for making purified products.
No, sir. It's like picking your poison, sir. The amount of dope. So when you think about addiction, sir, again, it's dopamine, Manna, sir. That's an increase.
Yeah.
So now, what are we trying to do is when a patient is on a opioid withdrawal, they need that dopamine level to increase. You know, like we take. I'll bring an analogy, sir. Like you take coffee or a chai or a coffee in the morning. That increases our dopamine. We have an addiction. So if it is increasing by 5%, I'm just giving you an analogy. Opioid is increasing the dopamine by 100%. So the craving is high. So the patient who's going a withdrawal during opioid syndrome, they require that stimulation of the brain, that amount of dopamine or monoamine to be released. So by giving cannabis under a particular dose, you still have the stimulation, but not with that level of addiction. The concept
so reducing.
Is, yes,
so
sir.
it helps gradual withdrawal.
Yes, sir. Yes, sir. See, like, so for people who are smoking, sir, they like to have the cigarette in the hand. They are not smoking, but having the cigarette in the hand psychologically is telling the brain you are having dopamine release. That follows the secondary mechanism with regard to addiction in the ventral tegmental region of the brain. So here, they are trying to give cannabis exactly, sir. And based on your approach, there's another one saying cannabis has been approved recently for epilepsy because they can decrease the neuronal fire. During the process of that, it's not only increasing the dopamine, the excitation associated with the withdrawal symptoms is associated with neuronal excitation. Cannabis can calm the patient, it can reduce the anxiety.
So it reminds
sir,
me
I
of
like that question, sir. But if you go to Varanasi or if you go to any Ganges region, you know, in our east, all the sadhus have been taking cannabis for a long time, but nobody died of that. That's, if you go from there, it's a natural product, which does not cause neurotoxicity. Addiction, yes, like coffee and tea. Yes, it is addiction, but it is not neurotoxic. Whereas opioid kills the patient.
It reminds me a story, you know, Dr. B.C. Roy.
Yes, sir.
once he was driving in a vehicle in Calcutta. So one person suddenly lied down in front of the vehicle. It was slow going in the crowded place. So he stopped the vehicle. Then that person told, he's please help me. He was seeking, look, but from he could read from his face, he was a chain smoker of the, in the Bengal, lots here also maybe use this. Instead of cigarette, they use bedi.
Really small, yes, sir.
Yes, that fellow was a chain smoker, continuous. But somebody advised him to stop totally. He stopped totally, but he became restless and he thought some other disease.
Yes,
So
sir.
he didn't ask a single question. He told, you take morning one and evening one bidis, you'll be all right. So it is like, using ganja as an antidote to recovery symptoms of the opioid addicts.
Yes,
That just
sir.
reminded me of that great story of this great physicians, Bishi Roy.
But
Thanks,
less
Murali.
devil is preferable.
Yes,
Absolutely.
always.
Absolutely.
Yes.
Like for alcoholics, sir. Like for alcoholics, they ask them
Yes.
to, if you go to the hospital, they have a drink. An alcoholic, when he comes to the hospital, they first give him a drink to calm him down.
Countries
Maybe.
have legalized the cannabis cultivation and use, but
Yes, sir.
we are very conservative.
Yes, sir.
Exactly.
Maybe low dose poison. Low dose is a medicine,
Yes, sir.
and high dose it is a poison. So any medicine,
Absolutely.
the dose
Absolutely, sir.
decides whether it is a medicine or a poison. So
You're right, sir. The dose and the dosage form. But whatever you have taught, I'm going back to your basics. We started from there. Dose and the dosage form, sir. That matters. You're right, sir.
yes. Okay. Thanks, Murali.
Thank
And
you, sir. Thanks, sir.
thanks all the participants who
Yes.
supported us throughout from first to the seventh webinar of arranged by Department of Pharmacy and University. Thanks all. Thanks all the organizing committee members, and of course our chairman, Professor and head department, Dr. Mohanto, and convener, organizing, what is our Donapal, the convener of this series of webinars. Thanks, everyone.
Thank you, sir. Thank you, sir.
Just a point.
Yes, sir. Yes, sir. Please, sir.
The Department of Pharmacy is known for organizing these continuing education programs for teachers, for professional pharmacists, and for others.
Yes.
And because of this COVID, we are not able to do, but our Dhanapal has taken this initiative to organize a series of, not just one, a series of webinars, and he requires all of our appreciation and applause.
Thank you, sir. It is because of all my teachers, colleagues, and other supporting staffs only we are doing. Thank you so much, sir. Now I request Dr. Devi to propose vote of thanks. Devi, please. Devi, you can start.
Sir,
Yes.
sir,
Yes.
audible, sir?
Yes. Yes. Ma.
Yes.
Yes.
Sir,
Yes, we are able to hear you.
Yes.
sir, good evening, everyone. Okay. Good evening, everyone. First of all, I would like to thank all our university authorities to give these opportunities to deliver the vote of thanks. I would like to express a sincere thanks to Speaker Dr. Muralishan Dhanasekaran, Professor, Harrison School of Pharmacy, Auburn University, the State of America, for making an excellent presentation and making this international webinar a very info and interesting. And also his excellent presentation on topic in very useful faculty and as well as students. Thank you, sir. I would like to thank our beloved professor and head of the department and chairman, Dr. G.P. Mantha, sir, for his wonderful support for this international webinar. And also I'd like to thank Professor Dr. P.K. Manna, sir, for his advice. And I would like to thank convener, Professor Dr. C.K. Dhanabal, sir. And also I would like to thank organizing members, Dr. S. Lomuthu Kumar and Dr. Ayurvedism's Associate Professor. Thank you, sir. And also I would like to thank Dr. D. Senthil Kumar and Dr. S. Sivakrishnan, Dr. G. Sivagama Sundari, assistant professors, and one of the organizing members. And also, finally, I would like to thank our international sponsors and thanks to all participants. Thank you, sir.
Thank you so much, Devi. Murali Krishnan, thank you so much. Yeah, I think you have to unmute your audio.
Thanks, sir. Thanks, sir. Thank you so much.
Even in the morning schedule, you have spent a lot of time, Murali. Thank you
Thanks,
so
sir.
much.
No, sir.
Your
The
parent
mind is
department-
fresh.
Yeah.
Yes.
Okay. Thank you, sir.
Thank you.
Thank you, sir. Thank you. See
Thanks,
you.
Murali. Murali, thanks.
Federation, you can close the session. Dr. Vendesh, able to hear? You unmute and talk. You unmute and talk. Sir, Kalacharman, sir?
Kalacharman has come, eh?
Yes, sir. That's what I want to.
Why
Kalacharman,
am I not able to see him?
unmute your audio. Kalacharman, sir, please unmute your audio. You unmute your audio. Kalacharman, sir?
First you close here.
We are closing, sir. Vendesh, close panning. You close it and leave the meeting and come in.
Venkateshan's audio is mute. He may not be able to hear you.
Yeah, I will call him, sir. That's it. வெங்கி மீட்டிங்க லீவ் பண்ணிட்டு நீங்க அன்மியூட் பண்ணிட்டு வாங்க திருப்பி எல்லாரும் வேணா பேசலாம் லீவ் பண்ணிடுங்க நிறைய பார்ட்டிசிபென்ட்ஸ் இருக்காங்க சரியான ஸ்பீச்